Now, before moving into the brainstem, I just want to review one more time some of the pathways in the spinal cord that we've talked about. Here we can see fasciculus grassus and cuneatus. Here we can see the zona lissar and the dorsal horn of the spinal cord. Right in here we'd have the lateral corticospinal tract. Lateral to that we'd have the dorsal spinal cerebellar tract. Down in here we'd have the famous ALS. Okay. I think that's about all the pathways we talked about. Now, as we move up into the brainstem, we're going to see some modification of these pathways. And this is our level 1. And what we can see here, this level is the decussation of the corticospinal tract, which we can see right here. So here you have a pyramid, and the cells of origin of these axons would be in the ipsilateral motor cortex. So here's an axon that's then going to cross and enter into a bundle right here called the lateral corticospinal tract. Now, after it enters the lateral corticospinal tract, it can just enter the spinal cord, and it's going to be right here. Okay? So here we're going to call it the lateral corticospinal tract, and we're going to say that if the cells of origin of these axons are in the contralateral motor cortex. Where are these axons going to terminate? They're going to terminate in the ipsilateral spinal cord. So any lesion in here is going to give you an hemiplegia, arm and leg, that is, weakness on the same side. Of course, you'd also have a Babinski and a hyper reflexes and things like that. But here your motor weakness and your spasticity is going to be ipsilateral. And a lesion down in here is going to be contralateral. Of course, if you interrupt this decussation here, you're going to be quadriplegic. If we move out into here, you can see our old friend, the ALS. And here, again, the cells of origin of these axons are going to be in the opposite dorsal horn. Not dorsal root ganglia, but dorsal horn. These axons are going to then go up the brainstem and terminate in the ipsilateral VPL. Now don't forget, what's descending in here are those fibers from the hypothalamus that are going to go down to the sympathetic outflow, T1 on down, T1 to L2. If we interrupt those descending autonomic fibers before they get to the sympathetic outflow, you're not only going to have a Horner's problem on the ipsilateral side, but you're also going to lose sweating in the head and the body on the same side. So here you're going to have a pain and temperature problem from the body on the opposite side and a constricted pupil on the ipsilateral side. We can see a little bit of the dorsal spinal cerebellar tract here. It doesn't show up really well on this level. Cells of origin of the dorsal spinal cerebellar tract would be Clark's column on the ipsilateral side, C8 to L3. If you just thought about the dorsal spinal cerebellar tract here, you'd say, well, the information in this tract is C8 on down on the ipsilateral side, unconscious proprioception. If I ask you where the dorsal spinal cerebellar tract fibers here are going to terminate, you'd say in the ipsilateral cerebellum. If I ask you how they get there, you'd say the inferior cerebellar peduncle. When we move up here, we get the spinal tract and spinal nucleus of 5. Now at this level, we're going to have axons that arise from the trigeminal ganglion on the same side, the geniculate ganglion on the same side, and the superior ganglia 9 and 10. And these axons are going to synapse here in the adjacent nucleus, spinal nucleus 5. They're going to then cross the midline and ascend. We can't really see a defined bundle here. They're going to cross the midline and ascend in the famous TTT, which goes all the way up through the brainstem to the VPM. Now don't forget, some of these crossing axons are going to go up the TTT and get off in the nucleus ambiguus on both sides. So don't forget that. So if we had a lesion right in here, we'd have a loss of pain and temperature from the ipsilateral side of the face. If we extended it into here, we'd have weakness on the ipsilateral side of the ipsilateral arm and leg. If we, let's take this lateral corticospinal tract and bring it down here, a lesion something like this. Here you'd have loss of pain and temperature on the opposite side of the body, constricted pupil on the ipsilateral side of the body, and weakness on the ipsilateral side of the body. Now let's do a gag reflex down here too, because remember, those fibers from 9 and 10 are coming down here, going to jump in the nucleus, cross and go up to TTT, and then go into both nucleus ambiguus. So if we had a lesion at this level and stimulated this side of the pharynx, that information would never get into the nucleus, wouldn't go up the tract, and wouldn't get into the two nucleus ambiguus. So if we had a lesion right here or here and stimulated this side, we wouldn't have a direct, we wouldn't have a consensual. If this was our only lesion and we stimulated the other side, we'd have both a direct and a consensual. If we move up here, we can see some myelinated axons here, fasciculus cuneatus. We can see some fibers here, fasciculus gracilis, and we can see a beginning of a nucleus here and here. So this would be nucleus gracilis, nucleus gracilis. Most of the fibers out here are alpha betas from dorsal root ganglia down to T6, okay? These would be dorsal root ganglia T7 and below. Also, there's some unconscious proprioception in here coming from those 1A, 1B, type 2 fibers that have come in, C7 and above dorsal root ganglia, that are traveling in the fasciculus cuneatus on their way to the very famous accessory cuneate nucleus. If we had a lesion in here like this, we'd have a loss of 2-point, etc., ipsilateral side, arm and leg. Have a lesion here, it's going to be pain and temperature from the face, ipsilateral. Lesion here is going to be pain and temperature on the contralateral side of the body, plus a constricted pupil on the same side. Now if we look at the next level, level 2, we see that a little bit of change. This probably is the most important thing. This is the pyramid. This is filled with thousands of cortical spinal fibers. Okay, now every level, rostral to the pyramidal decussation, we're going to say when we have a lesion of these cortical spinal fibers, we're going to have a problem on the opposite side, because remember, the decussation is just caudal to this. So if we had a lesion here, we'd have a contralateral hemiplegia, contralateral spasticity, contralateral Babinski. So these cortical spinal fibers have their cell bodies in the ipsilateral motor cortex. And then they're going to cross just caudal to here in the pyramidal decussation. If we move out into this area, again we know that the ALS is in here. Contralateral loss of pain and temperature from the body. Ipsilateral problem with the pupil and ptosis and things like that. This dorsal spinal cerebellar tract here is kind of tricky. It's heavily myelinated, so I think you should be able to identify it and know that it's carrying information about unconscious proprioception from level C8 on down. Right here, dorsal spinal cerebellar tract. It would have its cells of origin in the Clark's column, Clark's nucleus. It's going to use the inferior cerebellar peduncle to get to the cerebellum. We move up in here, you can see a good spinal nucleus right here, spinal tract 5. Again, at this particular level, you'd have cells of origin in the geniculate ganglion, the trigeminal ganglion, the superior ganglion of 9 and 10. They would come in here, jump in this little nucleus, cross, and go up the TTT. Beautiful, a beautiful fasciculus cuneatus, nucleus cuneatus, a slightly smaller fasciculus gracilis, nucleus gracilis right here. Coming out of these dorsal column nuclei, you see these little crossing fibers here called the internal arcuate fibers. They're going to come through here, cross, and join the medial lemniscus to the other side. So here you can see some internal arcuate fibers sweeping down here, going across and join the medial lemniscus. Now don't forget, if we had a lesion up in here, we're going to be ipsilateral loss at 2-point, etc. Okay? If we have lesions of the internal arcuate fibers, you're going to have an ipsilateral loss at 2-point, etc. Okay, we don't get that contralateral loss till we get over here into the medial lemniscus, which really shows best at the next level. So, we got the dorsal column nuclei here. We got the spinal tract and nucleus 5. We got a good dorsal spinal cerebellar tract. Now, where are those unconscious proprioceptive fibers? C7 and above, they're up here in good old fasciculus cuneatus, headed for our good friend the accessory cuneate in the next level. Now if we look at level 3, a lot of the things in here should be familiar, like the pyramids here form a very prominent bundle on the ventral surface. And here we have thousands and thousands of corticospinal fibers. Right above the pyramids, we're going to have the medial lemniscus. And these axons are destined for the VPL. They're arising from the opposite nucleus grossus and nucleus cuneatus. Right above the medial lemniscus, we're going to have a little TTT right here. Now, the TTT is comprised of axons that have arisen from the caudal spinal nucleus right here, okay? So then they're going to ascend, and the TTT is going to be right about here. So if you had a lesion of the TTT, you're going to have a loss, pain and temperature, from the opposite side of the face. Now if we come over here, we can see the spinal tract 5 and the spinal nucleus 5. Now one thing you should realize is that these axons that are descending in the spinal tract 5 are coming from the trigeminal ganglion and from the geniculate ganglion. And just at this particular level here, there are the superior ganglia 9 and 10. So they're going to have their ganglia right here, and their processes are going to join the spinal tract 5 right at level 3. So here you're going to have the pain and temperature from the pharynx coming into the spinal tract 5. And these axons are then going to descending in the spinal tract 5 down to reach the spinal nucleus 5. Okay? So think about 9 and 10 coming in here, descending down to the caudal spinal nucleus 5, and then eventually ascending here in the TTT. The axons then for the gag reflex can then jump off into nucleus ambiguus on this side and nucleus ambiguus on this side. What I'm trying to say is when we get rostral to level 3, we're only going to have axons in here from the trigeminal ganglion and from the geniculate ganglion, not from the superior ganglion 9 and 10. So when we're rostral to this and have a lesion in the tract, we don't have to worry about the gag reflex. Now we can move in here and we can take care of our good friend, the hypoglossal nucleus. There's cells in here whose axons go down like this and exit right here. So they run along the medial lemniscus here, and then they go between the olive and the pyramid to go out to the muscles of the tongue. Remember, these are lower motor neurons. So if you have a lesion in here, you're going to have atrophy of the tongue muscles on the ipsilateral side, and your tongue is going to deviate to the same side as the lesion. Now you can remember that these hypoglossal nuclei get cortical bulbar input and is primarily crossed. So if you have a lesion of the right, we'll just put it up here. If we have a lesion of the right motor cortex, you're going to lose that information down here into this hypoglossal nucleus, and your tongue is going to go to the opposite side. So think about a right motor cortical lesion. You're going to have a left hemiplegia, and your tongue is going to go to the left. Remember, the cortical bulbar pattern of the hypoglossal is really important. Move out here laterally, we got the good old dorsal motor nucleus. The key thoughts here would be preganglionic, parasympathetic, they go right out here, go out to a ganglia that's close to the organ, then you have a short postganglionic parasympathetic. The only thing we're going to have to remember from here is if you lose this, at least right after the lesion, you're not going to have anything working against the sympathetics, so the sympathetics are going to take over and you're going to have an increase in heart rate, which is called tachycardia. Now if we move down into our famous bundle here called fascicula solitarius, you can see fascicula solitarius has a partner here called nucleus solitarius. When we think about the rostral part of the solitary fasciculus and the nucleus solitarius, we're talking about taste. So here's some fascicles here that have their cell bodies in 3 ganglia. The geniculate ganglion comes in, anterior 2/3 of the tongue taste. The inferior ganglia 9 is going to come in here. That is, the central processes are going to come in here. That's going to be carrying taste from the posterior third of the tongue. And the inferior ganglion of 10, its central process is going to come in here into the fascicula solitarius. It's going to be carrying taste from the epiglottis. This taste information is then going to go into the nucleus solitarius, turns on that cell, and then there's a cell in nucleus solitarius that sends its axon up the same side all the way up to the thalamus in the solitariothalamic tract. Once it gets to the thalamus, it's going to be in the thalamic nucleus of the head, which would be the VPM. Remember, the VPL is for the body, VPM is for the head. Now, if we're in the caudal part of the tract and nucleus solitarius, we're only thinking inferior ganglia 9 and 10, not geniculate, inferior ganglia 9 and 10, and we're not talking about taste. We're talking about visceral afferent information from the gut. One of the best examples would be that the inferior ganglia 9 has a peripheral process that goes out to the carotid sinus, to those baroreceptors out there that pick up increase in blood pressure. Then the central process would come into the caudal part of fasciculus solitarius, jump into nucleus solitarius. When those baroreceptors start to fire more, those processes in those axons in cranial nerve number 9 coming in this caudal part of the solitary tract turn on these cells here in the solitary nucleus, and it says, well, the blood pressure is going up. Well, how can we lower the blood pressure? Well, a cell sitting in here can just jump right into the dorsal motor nucleus of 10. Then you've got a preganglionic parasympathetic fiber that goes out to the postganglionic, which will then go into the heart and lower the blood pressure. Now I'd like to talk a little bit about the accessory cuneate nucleus right here. It's lying just ventral to this big bundle here called the inferior cerebellar peduncle. The accessory cuneate nucleus is comparable to Clark's nucleus. That is, it receives unconscious proprioception, that is 1A, 1B type 2 fibers from C7 and above. So this unconscious proprioception comes into the accessory cuneate nucleus, synapses, and then the cuneo-cerebellar fibers, that is, they start in accessory cuneate, go up to the cerebellum, go through this peduncle here called the inferior cerebellar peduncle. So if you had a lesion of the accessory cuneate, you're going to lose unconscious proprioception from C7 and above. Now when we look here at this inferior cerebellar peduncle, we have to be aware of the fibers that are in this peduncle. Some of them are cuneo-cerebellars. A lot of them are dorsal spinal cerebellars that have come up from the more caudal levels. And of course the biggest component are the olivocerebellars that are crossing. If you have a lesion here in this inferior cerebellar peduncle, the overlying cerebellum on the same side has lost all this information. So you'll therefore have an ipsilateral incoordination of all muscles on the ipsilateral side. We can move down into this area right here where we have the ALS. We know that the cells of origin of these fibers are in a contralateral dorsal horn. So if we have a lesion here, we're going to have a loss of pain and temperature on the contralateral side of the body. Don't forget that they're descending fibers here from the hypothalamus. They're not going to get down to T1, to the T1 outflow, to the sympathetic outflow. So you're going to have a Horner syndrome on the same side, and you're also going to lose sweating from not only in the face, but from the side of the body, on the ipsilateral side of the body. The olive is involved in motor learning. We're going to learn more about the olive when we talk about the cerebellum. This is the inferior olive. This is a sole source of a fiber that goes to the cerebellum called the climbing fibers. So here's the olive. Here's a little bump that if we were looking at it from the outside, we'd call this the olive. We call this the pyramid. These are things we can see from the ventral and lateral surface. So the inferior olive projects through here like this, goes up into the contralateral inferior cerebellar peduncle, goes into the overlying cerebellum. So lesions of the olive will give you a contralateral incoordination of the arms and the legs. Finally, we have nucleus ambiguus here and here. And there's a little nucleus that sits medial to it called the inferior salvatory. The nucleus ambiguus is given rise to lower motor neurons that innervate soft palate, pharynx, and larynx. So they're going to travel with cranial nerve 9, 10. In the case of 9, it'd be the stylopharyngeus. 10 would be from the soft palate, or to the soft palate, levator veli palatini, and upper pharynx and larynx. So if we have lesions of nucleus ambiguus, we're going to have lower motor neuron damage, we're going to have atrophy, and the uvula is going to deviate to the strong side, not to the weak side, like the case of the hypoglossal. If we have lesions that kind of shade in here like this, all right, we're going to include this little nucleus here, inferior salvatory. Those are preganglionic parasympathetics that go out with cranial nerve 9 to the otic ganglion. So don't forget about this little nucleus sitting here. It's important to remember that the corticobulbar input to nucleus ambiguus is bilateral. So any lesions rostral to here that involve corticobulbars to nucleus ambiguus are not going to give any problems. Your uvula is going to be straight. Now I'd like to say something about the gag reflex. Here we have nucleus ambiguus right here, and the TTT would be right here. So let's do a couple lesions here. If we had a lesion in this TTT on this side, it's really going to prevent information coming from this side, okay? So if you stimulated this side of the pharynx, it'd come all the way down to the spinal tract and nucleus 5, cross and go up in this TTT. So if you had a lesion on this side right here in this TTT, if you stimulated this side, you wouldn't get a direct and you wouldn't get a consensual. But if you stimulated this side, you would get both a direct and a consensual. Now if we came out here and put a lesion in nucleus ambiguus on this side, if we stimulate this side of the pharynx, you wouldn't get a direct, but you'd get a consensual. If you stimulated this side, you wouldn't get a direct, but you would get a consensual. I'd also like to talk about the corticobulbars. Now remember, we have corticobulbars that are going to go into the hypoglossal and into nucleus ambiguus. And they're usually traveling with the corticospinal fibers. So if we had a lesion down here, these corticospinal fibers, the corticobulbar fibers that are coming down from the cortex that are headed for the hypoglossal and nucleus ambiguus have already gotten out of this system. So if we had a lesion here in the corticospinal system, we would not involve any corticobulbars because they have gotten out into the hypoglossal and into nucleus ambiguus, just rostral to level 3. So if we have a nucleus at the level of a lesion, for instance, if we have a lesion here and we're at the level of ambiguous and hypoglossal, you can always consider that the fibers have already gotten out into here and you won't have any corticobulbar problems. If we move up to level 4, we can cover some things that we already know. Here we would have the pyramids, the end of the pyramids. This is the beginning of the Pons coming in here. And you can see the remains of the pyramid, thousands and thousands of corticospinal fibers and corticobulbar fibers. Now, remember there are both corticospinal and corticobulbar fibers in here. So a lesion right here would give you a contralateral hemiplegia. Contralateral hemiplegia. But don't forget, we're rostral to the hypoglossal and to nucleus ambiguus. So we're going to be interrupting some fibers to both of those nuclei. Now remember, if you have a lesion here, you're going to have a contralateral hemiplegia. And the fibers to the hypoglossal are pretty much completely crossed. So the tongue will deviate towards the side of the hemiplegia. But this lesion right here, even though we're going to interrupt corticobulbar fibers to nucleus ambiguus, because this nucleus gets bilateral input, you're not going to have any problem with the uvula. So let's do it again. A big lesion here, cutting these corticospinal and corticobulbar fibers. Even though we're interrupting corticobulbar fibers to ambiguous and hypoglossal, the only problem we're going to see is in the tongue. So we're going to have a contralateral hemiplegia, and the tongue is going to go to the contralateral side. There won't be any what we would call lower motor neuron atrophy. We move up, we can see the inferior olive. We know that the olivo-cerebellar connection is crossed, so this would give us a contralateral incoordination. Here we have the medial lemniscus. We know that these axons are arising from the contralateral nucleus grassus acuneatus. These axons are going up to VPL, so we're going to lose 2 point, etc. Right above the medial lemniscus will be the TTT, which would give us a contralateral loss of pain and temperature from the face, head, and tongue. Right above the TTT would be the STT. Now, Don't forget, we're rostral to nucleus solitarius. So these fibers in the STT are ascending, just like the TTT, to VPM. And if we have a lesion in the STT right here, we're going to lose taste on the ipsilateral side. We know the ALS is in here, and we know the descending autonomics are in here. So we're going to have an ipsilateral Horner's, no sweating in the head and below on the body. And we're going to lose contralateral pain and temperature from the body. This would be the spinal tract, spinal nucleus. Now remember, the only thing in here now would be fibers that are descending to the spinal nucleus from the trigeminal ganglion and the geniculate ganglion. The fibers from the superior ganglion 9 and 10 intercaudal to this at level 3, so we wouldn't interrupt or have any problem with the gag reflex here. Also, if we go over to the TTT, any lesions of the TTT are not going to give you any problems with nucleus ambiguus. You're just going to lose pain and temperature from the contralateral side of the head, face, and tongue. But remember, nucleus ambiguus is caudal to this, so the fibers in the TTT involved in the gag reflex have already gotten off. So you're going to have the gag reflex, you're just not going to feel that pain and temperature from the pharynx. The key identifiable structure here at level 4 is this big inferior cerebellar peduncle. And it's draped by the dorsal and ventrocochlear nuclei, and on the medial side, the vestibular nuclei, right here. So you can see this level is cranial nerve 8. Cranial nerve 8 would have its cell bodies out in the cochlear ganglion, vestibular ganglion. The auditory part of 8 comes into the cochlear nuclei, dorsal and ventral. The vestibular part comes medial, comes up in here medial to the vestibular nuclei. So let's take care of this. What are the fibers in here? We'd have the cuneo-cerebellars, dorsal spinal cerebellars, and the olivocerebellars. This bundle's getting ready to go up into the cerebellum. We will never see this bundle again in our series. Again, if you have a lesion here, you're going to lose all the information to the overlying cerebellar hemisphere, and you know that one side of the cerebellum controls the same side of the body. So if we have a lesion here in the inferior cerebellar peduncle, we're going to have incoordination on the same side. Lesions in the cochlear nuclei or in the nerve, you know, give you deafness. Now, the vestibular nuclei. Again, if I was to ask you where the cell bodies are of the axons that end in here, you'd say Scarpa's ganglion or vestibular ganglion out here in the more periphery. The vestibular part of the 8th nerve comes in synapses in the vestibular nuclei. Now, you know that there are a couple pathways that go out of these nuclei and descend, go down to the spinal cord, and help us maintain our balance. And this would be the lateral vestibulospinal tract, which arises from the lateral vestibular nucleus, and the medial vestibulospinal tract that arises from the medial vestibular nucleus. But they're going to descend, and the lateral is going to descend completely uncrossed and travel down in the ventral funiculus and help us maintain our balance. And the medial vestibular spinal tract is going to descend mostly uncrossed, but also crossed. And you can actually see the medial vestibulospinal tract right here. Okay, so there's cells in the medial vestibular nucleus, mostly uncrossed, some crossed, that descend in this position in the brainstem, go down eventually into the ventral funiculus, and go down primarily to the cervical level. So that helps you control the position of your head, you know, as you start to stumble or whatever, to try to keep your head upright. So this would be the medial vestibulospinal tract. Right underneath it would be a little circle called the STT, then another little circle called the TTT, and then the medial lemniscus. Now, we have lesions in the vestibular nuclei right here. Let's just say that this one is wiped out. This one is controlling. So this complex has a lateral vestibulospinal tract and a medial vestibulospinal tract. So this side is much more active, and you're going to tend to stumble towards the weak side. So if we have a lesion on this side, and this is dead, and this is very active, you're going to stumble towards this side. Now don't forget, this complex is also connected to the eyes muscles, okay? And you got to think about the fact that if you lost this nucleus on this side, sure, you're going to stumble this way, but this complex on this side is going to slowly drive your eyes this way, and then it'll snap back fast. And we name the fast direction the nystagmus, all right? So if this is dead, this one drives your eyes slowly to the way that you're stumbling, but then snap back. Your eyes, the actual nystagmus will be in this direction. So here's your lesion. This one's active. Eyes go slowly this way and snap back. So to the lesion, you stumble towards the lesion. The slow movement of the nystagmus is towards the lesion, but the fast movement is away. Now if we turn to level 5, now we're in the middle to caudal third of the pons. We still have our thousands and thousands of cortical spinal fibers down here and cortical bulbar fibers. We have something called the basilar pons down here. And then we have something called the tegmentum of the pons up here. These are our big middle cerebellar peduncles. Now, the inferior cerebellar peduncle is gone. It's already gone up into the cerebellum. So here we have a really typical section of the pons, middle cerebellar peduncle, basilar pons down here, tegmentum of the pons up here. Here's your other middle cerebellar peduncle. We can see these thousands and thousands of cortical spinal fibers. Also mixed in here are cortical bulbar fibers, and then there's some other fibers in here that are getting off in the basilar pons here that we call cortical pontine fibers. Let's take the cortical spinal fibers first. We have a lesion down here of these cortical spinal fibers. It's going to have a contralateral hemiplegia, a Babinski, and all those other things, increased reflexes and spasticity on the contralateral side. Now there are also cortical bulbar fibers in here, and you know we're rostral to the hypoglossal and to the nucleus ambiguus. So we're going to interrupt those cortical bulbar fibers to hypoglossal and ambiguous. We're not going to have to worry about the fibers going to ambiguous. The uvula is going to be straight because it gets a bilateral input. We are going to have to worry about the fibers in here, the cortical bulbar fibers in here that are headed for the hypoglossal, because they're going to be crossing over to the other hypoglossal. So if you had a lesion of the cortical spinal fibers in here, you're going to have contralateral hemiplegia. You're going to interrupt the cortical bulbar fibers in here to ambiguous and 12, but you're only going to have to worry about 12. And the tongue will deviate towards the same side as a hemiplegia. Now you've also got these millions and millions of cells in here called pontine gray cells. These are pontine gray cells. And they're getting their information from cortical pontine axons that have started up in the cortex of the same side, right alongside those cortical spinals. Now if you think about it, these cortical spinals are going to influence the other side of the spinal cord, all right? So think about a motor signal coming down these cortical spinal axons. It's actually going to tell the opposite side of the spinal cord what to do. So the cortical pontine fibers are also carrying the same signal that's being carried by these cortical spinals. So the cortical spinal fibers are going down, they're crossing, telling the lower motor neurons what to do. The cortical pontine fibers that are traveling in here get out and tell all these millions of pontine gray neurons about the signal being sent down the cortical spinal tract, like move your arms 12 degrees and your legs 12 degrees. But these cortical pontine axons that are traveling in here and getting out on all these millions of cells is carrying the same signal that the cortical spinal fibers are carrying down to the spinal cord. So this signal was told to all these millions of neurons here, and then there are cells in here which have axons that cross the midline like this and go up into the contralateral cerebellum. And we call these axons ponto, from the pons, pontocerebellar axons. Okay? So now you know that these pontine gray neurons are getting a signal from the ipsilateral motor cortex about the movement to be carried out. Cortical spinals will carry out the movement. The cortical pontines will tell the pontine gray about the movement. The cell will then send its axons across up the middle cerebellar peduncle and tell the opposite cerebellar cortex about the movement that's being designated by this motor cortex on the other side. So cortical pontine, pontocerebellar. Now, you'll remember that this cortical spinal fiber is telling this opposite side of the spinal cord what to do, and when the movement starts to happen, the dorsal spinal cerebellar tract and the cuneo-cerebellar tract start sending information up the same side, remember they're uncrossed, to tell the cerebellum about what is really happening to those muscles. So what is happening in this cerebellum this side is, it's getting a signal about what is supposed to happen from these pontine gray neurons, which are in turn getting their information from the cortex. So what's being compared up here is what is supposed to be happening in the motor program, and the dorsal spinal cerebellars and cuneo-cerebellars are telling the cerebellum, you know, this is what really is happening. So there's a comparison between what the motor cortex wants to do and what the muscles are actually doing. So some comparison takes place in the cerebellum, and then a signal is sent out of the cerebellum that will eventually take care, tell the cortex and the red nucleus to kind of fix or change the program if it's not matching up with what is actually happening from the muscles. So the signal comes down, do this. The muscles say we're doing this. Cerebellum calculates the difference, sends the signal out via a pathway we're going to talk about pretty soon in the next level, through the superior cerebellar peduncle. It can then go to pathways that will correct the movement very fast. It'll also go back up the cortex to tell the cortex how the movement is off. So let's just take a little lesion of these pontine gray neurons here. If you just went in here and selectively with a virus wiped out all these pontine gray neurons, the opposite cerebellum has lost important information. And you know that this cerebellum controls the same side. So a lesion of the pontine gray will give you a contralateral incoordination. However, a lesion of this middle cerebellar peduncle right here is going to interrupt all the axons going up into this cerebellum. So if you have a lesion of the middle cerebellar peduncle, you're going to have an ipsilateral motor incoordination. So the peduncle has its cells of origin. Cells of origin of these axons are in the opposite pontine gray. A lesion in this peduncle gives you ipsilateral motor incoordination. A lesion in the pontine gray is going to give you problems in coordination on the opposite side of the body. So don't forget, down here, basilar pons, cortical spinals, cortical bulbars, cortical pontines, pontine gray cells in here that have pontocerebellar axons that fill this peduncle. Look how much bigger this peduncle is than the inferior cerebellar peduncle. If we move up a little bit, we've got some friends from earlier things in the auditory pathways. We have something called the lateral lemniscus right here. Now you'll recall that this lateral lemniscus can have some of its cell bodies here in these cochlear nuclei. Remember the dorsal and ventral, and we traced the pathway up from the ventral cochlear nucleus that went up the lateral lemniscus. Going up the lateral lemniscus is going to go all the way up to the midbrain, to the inferior colliculus. It's going to synapse in the inferior colliculus. Inferior colliculus will project to the medial geniculate. The medial geniculate will project to the auditory cortex. So here's some of that lateral lemniscus. Now you also recall that some of the axons in the lateral lemniscus could get off into something called the superior olive. So here, this light structure right here, this nucleus right here is your superior olive. So axons can either keep going up in the lateral lemniscus to go up to the inferior colliculus, or some can get off and go into the superior olive and synapse. Now the superior olive could do a lot of things, but I think the one in the course book is we have the axons that cross the midline and go up the other lateral lemniscus. So this superior olive could have axons that cross and go up in the other lateral lemniscus. So let's take this superior olive axons and its crossing. There's a big bundle here in the midline, which we call the trapezoid body. Okay, so actually a lot of this is a trapezoid body, these crossing fibers here. So above these fibers we have the tegmentum of the pons, and below the fibers we have the basilar pons. So again, here's the lateral lemniscus. If we had a lesion in the lateral lemniscus, we can just say subtle auditory deficits. If we have a lesion in the superior olive, we can say subtle auditory deficits. But we know that axons arising from cells in the superior olive cross the midline, and if it's crossing the midline like this, it's got to be the trapezoid body. And then the axon can continue to the other lateral lemniscus and take on up. So here would be the lateral meniscus on this side, the superior olive, the trapezoid body, superior olive on this side, right down here, lateral meniscus. So there's a lot of other things in here though. There are a lot of other things in here besides fibers that are crossing from the superior olive. But if I'd point to it right here on the midline, it's got to be the trapezoid body because these fibers are crossing the midline. Now embedded in here with some of the superior olive axons are some of our most important players from earlier levels that are ascending, but they're becoming compressed together. So what we're going to have in here is the TTT, trigeminal thalamic tract. We're going to have the ALS in here, and we're going to have the medial meniscus. So they're all jammed in here together now. Up in here would be the STT. So it's not in here. What's in here is the ALS, the TTT, the medial meniscus, and some trapezoid body fibers. Now, your problem with the TTT is going to be contra. Your problem with the medial meniscus is going to be contra. Your problem with the ALS is going to be contralateral loss of pain and temperature. But don't forget those fibers that are descending down to the sympathetic outflow from the hypothalamus. So your pupil problem is going to be IPSI. So contralateral loss of pain and temperature from the body, ipsilateral pupil problem, contralateral loss of pain and temperature from the head, face, and tongue, and loss of 2-point, etc. from the contralateral side. So now these are all kind of converged together and makes it a little bit easier. But the STT is not in here, so you're not going to have any taste problems. Now if we move up in here, not to be confused with this superior olive, is the motor 7. Motor 7. This is a tremendously important cranial nerve. This is motor 7. There are lower motor neurons in here that are going to go out to the branchiomeric musculature of the second arch. So these are lower motor neurons that are going to go out to a lot of facial muscles and things, the stapedius muscle, orbicularis oculi, to help you close your eye. So these are lower motor neurons in here, and they take a rather circuitous route out of here. So here be your motor 7 sitting right here. There's a cell in here. Instead of just jumping out and going on out, it goes medial and goes up and over this nucleus here, which is the abducens nucleus. So here's a motor 7 neuron. It's got an axon that goes up and over, and then you can see it right here coming out. Okay? So motor 7, muscles of facial expression, up and over and out like this. And here's the abducens nucleus, which we're going to talk about in a minute. And the fibers would go right over the top, and you have a little bump here called the facial colliculus. Here's the abducens sitting right here. You could do the same thing by having a little lesion of these rootlets here. So you have a lesion in the nucleus, or when it goes over the top here in the facial colliculus, or cut them right here, okay? Now, if you have a lesion in here or in these rootlets right here, you're going to have problems with muscles of facial expression. You're also going to have something called hyperacusis, which is sensitivity to sounds. Your orbicularis oculi is going to be fouled up, so you won't be able to close your eyelid. So these are some problems with motor 7. Now, medial to motor 7, right here, it's kind of like the situation with nucleus ambiguus. Nucleus ambiguus is in the medulla, but it innervates branchiomeric musculature, and it has a nucleus medial to it called the inferior salvatory. Now when we're up here in the pons, we have lower motor neurons that innervate branchiomeric musculature of the second arch. There's a little nucleus medial to it called the superior salvatory. So here we have superior olive, so you might help you remember superior salvatory. Now, these axons are going to go out with cranial nerve 7. And they're going to go to the submandibular ganglion, which in turn then has short postganglionics to submandibular and sublingual glands. They're also going to have some cells in here. Sometimes we call it not the superior salvatory, but we can also call it the lacrimal. They're going to go out, and they're going to go to the pterygopalatine ganglion and then go to the lacrimal gland. But the most important thing is that these preganglionic parasympathetic cell group fibers here associated with 7 do not loop up and over. They do not go over the facial colliculus, form part of the facial colliculus. Instead, they just sneak out this way, like this. So here you'd have your preganglionic parasympathetics. They just loop like this. They could also go like this. But the lower motor neurons in motor 7 actually go up and over and form part of this facial colliculus. We have a little bit of spinal tract here, spinal nucleus. At this particular level, these axons would contain processes that have come in from the geniculate ganglion for 7, and of course we have the processes coming down from the more rostral levels of the pons that have their axons from cell bodies in the trigeminal ganglion. But of course, there would not be any axons in here from the superior ganglion of 9 and 10 for the gag reflex. Don't forget also that the TTT down in here, if you have a lesion of the TTT down in here, you're not going to have any trouble with the gag reflex because those axons have already gotten out in nucleus ambiguus, which is a couple levels down. If we move into this little area right here, we have something called the paramedian pontine reticular formation. We can also see the rootlets of cranial nerve 6 coming out here. We can see abducens nucleus sitting right here, and we can see the medial longitudinal fasciculus right here. Now, as you recall, hopefully, the paramedian pontine reticular formation is going to get information from the opposite vestibular nuclei and from the opposite frontal eye fields. And what's important is that these paramedian pontine reticular formation neurons feed into the abducens nucleus. So PPRF feeds into abducens on the same side. Now you know that the abducens nucleus, if this is on the left side, then has cells in it, some of which are lower motor neurons. And those cells have axons which come down like this and go out. So these little rootlets here would have their cell bodies in abducens neurons, lower motor neurons, of the same side. Now, and of course, this pathway here will make your ipsilateral eye go laterally. So in other words, if this is the left abducens, these rootlets will go out to the left lateral rectus and cause your eyeball to go laterally. Now, there are also small cells in here, interneurons. An interneuron is a type of cell whose axon does not leave the central nervous system. So these little interneurons in here have axons which cross the midline and go up the medial longitudinal fasciculus all the way up to the oculomotor complex. So this MLF is going to be with us for a ways. Go all the way up to the oculomotor complex, and particularly to the medial rectus neurons, So this nucleus then has lower motor neurons whose axons go out and supply the ipsilateral lateral rectus, also has interneurons whose axons go up the opposite MLF and innervate the medial rectus neurons of the opposite eye. So you can see if you stimulate this nucleus here, your eyes are going to go ipsilateral. They're going to go to the left. This will go left, and then this will make the opposite eye go medially to the left. So if you stimulate this left nucleus, both eyes are going to go to the left, or towards the ipsilateral side. Now, the same thing will happen if you stimulate the paramedian pontine reticular formation. If you stimulate this, it's going to turn on abducens. Abducens is going to turn on these big axons that go out to the lateral rectus. It's also going to be turning on the small neurons whose axons go up to the opposite eye and have it turn medially. So if you stimulate the PPRF, your eyes are going to go ipsilaterally. If you stimulate the abducens nucleus, both eyes are going to go to the left. Stimulate here, both eyes will go to the left, or ipsilaterally. Now, if you stimulate these axons right here, only the ipsilateral eye, or the left eye, is going to go laterally. So what would happen if you had some lesions? Well, if we had a lesion in the PPRF, we couldn't tell the abducens to fire, so we couldn't turn our eyes, both eyes, together in this direction. So you couldn't get your eyes to go ipsilaterally, or in this case, you couldn't get both eyes to go left. The eyes would be aligned, but you just couldn't get them past the midline to the left. And in fact, because the other PPRF is working, the eyes could drift a little bit to the right. Now, compare a lesion that would get this PPRF on the left and these cortical spinal fibers on the left. If you think about which way your eyes might be drifting, they're going to be drifting. Remember, you can't turn them this way. They're going to be drifting a little bit to the opposite side, to the right. You think about a lesion of the cortical spinal fibers here, where's your hemiplegia? Well, it's going to, if these are the left cortical spinal fibers, it's going to be on the left side. So in a case of a lesion of the PPRF, the eyes are going to drift a little to the right. The hemiplegia is on the right, so we say that the eyes are looking towards the hemiplegia. Now, if you had a lesion of the abducens nerve here, you're thinking now, this is just, you're only going to have problems with one eye. Now, when one eye is affected, when one eye deviates, you have double vision or diplopia. When one eye is affected. Now, that's why you don't get diplopia with a lesion of PPRF, because both eyes are having the problem, and so your eyes are still aligned. They're not looking at two different points. But if you have a lesion of just these rootlets here, the left eye is going to be deviated medially. It's going to be deviated a little bit medially, and the two eyes are not going to be looking at the same spot. So what you want to do is you want to rotate your head toward the lesion. If you rotate your head towards the lesion, the other eye, the right eye in this case, will move laterally and then match up with which way the left eye is looking. So let's do that again. Your left eye would be deviated medially. Your right eye would be straight. If you turn your head to the left, your right eye will just rotate out naturally and match up with the left eye and get rid of the deviation or the double vision. So a lesion of the left abducens nerve, you will rotate your head to the left to get rid of the double vision. If we move up into the abducens nucleus, what would happen with a lesion here? Again, you can't move your eyes towards this side. You can't get them across the midline to the left. Now, it's just like, again, you're going to kill these lower motor neurons in the left nucleus, which gives rise to these rootlets. So your left eye is going to be deviated a little bit to the right. Now, you've also lost some input over here to this right medial rectus. So the right medial rectus is going to be weak, so the right eye is going to be deviated a little laterally, but the left eye is going to be deviated more medially. So to match up the two eyes, you're going to, again, just like in the case of the nerve, rotate your head towards the weak abducens nerve, so that would be to the left. So in this particular case, when you just have the double vision from the weak lateral rectus muscle, left side, you would rotate your head to the left. In this case, again, you would rotate your head to the left. Now, in this particular case, you would have atrophy of the left lateral rectus muscle. In this particular case, you'd have atrophy of the left lateral rectus muscle, but only weakness of the right medial rectus. So we know that there are large and small cells in here. The large ones send their axons out the nerve. The small ones send their axons up the MLF. So what would happen if we had a lesion here of the MLF on the right side? Well, what couldn't you do? You couldn't get your right eye to turn medially when you try to look to the left. Okay, remember, this complex will get your eyes, both eyes, this eye and this eye, to turn to the left. If you've lost this drive, when you try to look to the left, you can't get that opposite medial rectus to turn on, to look to the left. Okay, so this eye also would be deviated a little bit laterally, because you've lost that drive on the medial rectus. So if your right eye is deviated a little bit laterally, you want to turn your head towards the weak eye, turn the right, so that your left eye moves a little bit laterally and you get rid of the double vision.